Okay, so I would like to begin thanking Cesar for the kind presentation that he made of me. So I'm going to present today the results of an analysis that I've been doing here since I arrived in Minnesota half a year ago, making epidemiological sense out of large data sets of PRRS sequences. So I feel like this slide is a little bit preaching to the choir a little bit, so I'm going to move a little bit fast on it. But, well, PRRS, reproductive and respiratory syndrome, the estimated annual impact in the U.S. swine industry is around $600 million. We are working with data of the MSHMP project, so we have a collection of sequences that are sent to us of PRRS. This seems to be an important tool to investigate epidemiological patterns of occurrence of that disease. This is not something that we are first trying to do. There are similar approaches that have been done investigating influenza in humans, for example. And the general idea that we're trying to do here is trying to use this sequence information that we are getting from the systems via the MSHMP project and trying to relate the occurrence of those sequences to systems, to the type of farms, to the type of animal movements, or to try to understand patterns that might be driving the spread or the occurrence of those different PRRS sequences. So we're working with 1,900 PRRS sequences that were sent to us via systems participating in the MSHMP projects. We are only using data from 2015 to 2017, even though we do have data that expand backwards in time and a little bit further in time also. We have information on the farm, or the specific farm in which that sequence appeared, the system, the production type, the location, and the date in which that sequence was obtained in that specific farm. Each farm was tested based on animals' clinical suspicion. However, we do not have data on negative farms, so the data set that we're working with is only the positive ones. So there might have been, or there were, farms in which the attending veterinarian suspected of a possible PRRS, sent that sample for sequencing, the sequence was negative, and I do not have information on that, even though I do know that that does happen. But this is important because it limits us a little bit into calculation, for example, incidence. So this is an exploratory analysis. We're trying to investigate genetic similarities between sequences in different points in space and time, and try to understand how that could provide insights for understanding PRRS transmission. So the objectives of my analysis here is to stratify the MSHMP sequences into different genetic groups, also to describe temporally and spatially the occurrence of those PRRS genetic groups, to evaluate if the frequency in which those genetic groups occur is similar between different years, systems, or production types, and to evaluate if movement of animals contributes to the spread of those specific PRRS genetic groups. So this first slide here is my first result slide. So again, 1,900 sequences. Those sequences were distributed over 750 approximately unique farms, and by unique I mean there were 750 different farms that reported sequences to us. Those farms were from different production types. So I'm working here with a subset of the MSHMP data, so I'm only using here 3 systems. One of those systems contributed to approximately 65% of the data that I have, while the system that less contributed to the sequences I have contributed to 9% of the sequences. This map here shows the distribution of the sequences stratified according to the systems, so different colors here represent different systems. And this figure to the right shows the temporal distribution of the sequences that were reported to us, stratified by months. So we have, again, data from 3 years, 2015 to 2017. 40% of our data were from the first year, and 25% of our data was from the last year of the time series. Next, we try to cluster those sequences into different genetic groups, and this can be a little bit tricky. Before I enter into more details into how we proceeded into doing so, we first removed duplicates, and by duplicates I mean different sequences that had different reports of sequences that had the exact same nucleotide composition. So they were 100% similar. and also recombinant sequences. Additionally, I also obtain extra sequences, so I obtain the Lelystad and the VR2332 reference sequences. I also obtain 3 vaccine sequences and approximately 840 what I'm calling anchor sequences that I'm going to use to classify those sequences into 9 different lineages. So I'm working with the papers that were published by Shi in 2010 and 2013. And in this paper, they do this kind of classification of PRRS into lineages, and this is where I obtain my anchor sequences. So this figure here in the bottom part of the slide shows that I begin my analysis with 1900 MSHMP sequences. I dropped approximately 500 and because they were duplicated, and approximately 10 because they were recombinants. I also had 5 prototypes or vaccines and 840 anchor sequences for a total of 2,200, approximately 2,200 different sequences. So based on the anchor sequences that I obtained from the paper that I just mentioned, I try to classify the MSHMP sequences into those different lineages. And this is what this image does. So here I'm trying to show what is the minimum genetic distance of each of by ours I mean the MSHMP sequences, to the closest anchor sequence that I had in my anchor database. So I was able to obtain or to classify our MSHMP sequences into 4 different lineages: lineage 1, 5, 8, and 9, and 83% of our sequences were classified into a single lineage, lineage 1. Also, there was this group here that I labeled poor classification, because the average minimum distance to an anchor was approximately 40–45%, so I was not confident that the lineage that was assigned to those sequences was correct. So I relabeled those as poor classification, and I'm going to try to further stratify those later on. So in this slide, I'm trying to show how did the phylogenetic tree that we constructed look like. So between different shades or hues of color, so among the red, there's the red or the dark red and the pink. So the darker colors are sequences from the MSHMP project, and the lighter colors are anchor sequences. So again, here I'm just trying to show that 83% of our sequences, which is this huge dark red group here and also here, were classified under this lineage 1 lineage. So this is a little bit of a problem for me, because if I'm trying to look for patterns of occurrence of those different lineages, having 83% of my observations clustered into a single cluster group does not give me a lot of power to look for patterns of occurrence of that group. So using this software called Cluster Picker, which also looks for genetic distance, I try to further stratify that lineage into different groups. And what this software returned to me was that the software was able to stratify this lineage 1 into what I'm calling now lineage 1A, B, and C. Lineage 1 was still big, but it was a little bit smaller than it was before. And I also used the same software to further stratify the poor classification one. Even though I didn't have any anchor sequences that were close to those, not all sequences inside this poor classification group were similar to each other. So I was able to stratify this poor classification group into 2 different groups, what I'm calling here type 1 alpha and type 1 beta. So in the end, I ended up with 8 different genetic groups: lineage 1A, B, C, lineage 5, 8, and 9, and type 1 alpha and beta. I debated a little bit about adding an RFLP patterns most commonly found in those specific lineages, because I'm not using RFLP to stratify my sequences, but I figured out that it would be useful for practitioners that may sometimes use RFLP patterns in their day-to-day activities. So this lineage 1A, which was the most common lineage in the sequence that we were analyzing, 64% of the sequences inside this lineage were 174s, and the second most common were others. I mean, sequences that were not 174, 252, 144, and so on and so forth. So here, everything that is a black dot means 0%. I just replaced 0 just to avoid clutter in the slide. So the most common RFLP for L1A was 174, for L1B was 118.2, for L1C was both, well, more 144, but also 134, and so on and so forth. This next slide here, I'm showing the spatial distribution of those genetic groups over space in the area that we were working. So it's a little bit hard to see any clear patterns here. The distribution of the different genetic groups seems to be kind of even in this area. Here I'm trying to plot the occurrence of those different lineages or genetic groups over time. So you can see that there are some lineages that seem to occur more often in specific parts or in specific times in my data series, and then they decrease over time. Other seems to appear in specific moments. I'm going to go into further detail into that. So right now, this slide, or this and the next following two slides, they try to explore that. So right here, I'm trying to plot the distribution of those genetic groups over space and time. So I'm going to go forward 2 slides and then backward, just because what I'm showing those different slides is the year difference. So this was the distribution in 2015, 2016, and 2017. So if you look in the colors of the dots that are occurring in those different years, you can see, for example, in 2015 there were more blue dots, which is the lineage 1A. This is what I highlighted here in the bottom left. So 75% of our sequences in the first year were from this lineage, or this genetic group, while in the last year those were 57% of the sequences that were reported. On the other hand, for example, if you pick type 1 beta, this was 0.1% of the sequences reported in the first year and grew to be 6% of the sequences reported in the third year. So the frequency of those different strains or genetic groups that we identified in this area here is not equal in different years. So even when using a genetic classification system that perhaps does not allow for such variations, such as RFLP, for example, we're still able to capture that kind of information. Here I stratified the data. Instead of stratifying it over time, I stratified it across different systems. So there are 2 interesting things that I like to point in this slide here. There are 2 possible comparisons that I like to make. The first of those is, if you look within a single system, let's say system A over here, you can try to understand what is the pattern of occurrence of those different genetic groups over time inside or within that system. And for example, for this system A, nothing very clear appears, but if you look at system B, you can see that this red genetic group, which is lineage 1C, was kind of rare in the first year, and then it started to increase. And if you sum the red one, which is the lineage 1C, plus the yellow one, which was lineage 5 in the last part of the last year, it was more than 50% of all sequences reported in that system. Another thing that is interesting to look is not look within systems, but try to make comparisons between systems. So this pattern that we are observing here in system B is not the same that we are observing in system A, and not the same that we are observing in system C. So even though the systems are close to each other, there are different patterns across different systems. The same thing can be done stratifying the data according to the production type of the farm that originated the sequence. The patterns are a little bit harder to see, but there are some interesting things that I would like to point out. So for example, lineage 5, which is shown as the yellow bars on the graphics, it was responsible for 17% of our sequences that were reported in finisher farms and 19% of the sequences that were reported in nurseries, but only 7% of the sequences that were reported from sow farms. On the other hand, if you look, for example, for type 1A and type 1 alpha and type 1 beta, those 2 sequences, they were in finishers and nurseries, they were 0.7 and 0.5%, 1.5 and 1%. And if you look for sow farms, it was 3 or almost 4% and almost 3%. So again, this also shows that the distinct production types have different occurrences of those different genetic groups. We then try to analyze, try to find what could be the possible contribution of movement of animals and the occurrence of those specific genetic groups. Unfortunately, we only have data for specific systems, so we do not have data for movement of animals for all systems. And this is also a point that I would like to make. The presentation that I'm making here is a little bit of an exploratory analysis. We're going to be presenting later this afternoon on another session by Kim van der Waal some further analysis regarding the usage of movement data into the prediction of occurrence of breaks of PRRS and PED. So I would like to make a point here. If the system that you are working with, you're not collecting movement data, please consider doing so. It might be able to provide interesting layer of data for analysis, and in the end, it might be important if we if we end up with good results in the prediction project. It's important to be able to pick point-specific movements that represent higher risk of transmission of those diseases. The whole idea of trying to analyze movement in this data set is that movement is not equal, movement of animals is not equal among all farms. So some farms link more often with other farms, and if movement is responsible for transmission of those diseases, then I would expect to see sequences that are more similar between farms that link more often. And this is what this figure here shows. This figure is trying to express the strength or different communities of movement between different systems. I only have movement data for 2 systems, so this bottom blob here is one system, and this other blob here is another system. So for this bigger system here, you can see that there are different sub-communities of movement of animals inside that system. Another thing that is interesting to note here is the great number of connection links that are made between different farms. Perhaps the image is not very clear, but there are several edges or several lines that connect those different dots, and this is what the point I'm trying to make. Specific farms links to several other farms, and this is important if we're trying to understand the occurrence or the spread of those diseases over movement. So we used a statistical test to evaluate the occurrence of cases, if the occurrence of cases of a specific genetic groups is a result of propagation of pathogens through network links. And this is what this figure shows. So it's a similar way of plotting the farms as the previous figure. But right now in red are being shown farms that were positive for lineage 1A. In blue, it might be harder to see, but in blue are farms that they reported sequences but not from this specific genetic group. And in gray are farms that we know movements happened, but we didn't get any report of sequences from those farms. So we run this test, and this test returned us what is the significance of the movement into a possible transmission of those pathogens through network links. And what I'm going to advance here a little bit, but what this data shows is that we see evidence of transmission of specific genetic groups through movement links. However, this is not a rule for all genetic groups. So, highlighted in red cells here are the specific moments and the specific genetic groups in which we did observe a significant contribution or a significant suggestion that movement could perhaps be responsible for the transmission of those genetic groups. However, if you look for type 1 alpha and type 1 beta, we never see contribution of movement to the spread of those genetic groups, which is interesting. It's a little bit hard to explain. If you remember, type 1 alpha and type 1 beta were sequences that were more commonly found in sow farms, so it's hard to explain. Those sequences, those genetic groups are happening in sow farms, but are not being moved away from those sow farms. Again, movement of animals seems to be associated with occurrence of certain PRRS genetic groups. There we see a similar pattern when data is stratified by year. And again, a limitation of this data is that we only have this data available for specific systems. Last, we decided to explore a little bit more on the phylogenetic tree of the sequences that we had. And I mentioned in the beginning of my presentation that I obtained 2 reference strains and also 3 vaccine, not strain sequences and 3 vaccine sequences. So I decided to plot where those references and those vaccines are located inside my phylogenetic tree. So this is the Lelystad reference sequence. This is the VR2332 reference sequence. And this is where the vaccine 1 is located, vaccine 2, and vaccine 3. And one of the things that called our attention was that the PRRS vaccines are not from the same genetic group as the most prevalent sequences that we observed here. So the most prevalent sequences or lineage is this big lineage here, lineage 1. None of those vaccines that we looked for are from that specific group. PRRS vaccines are known for limited heterologous protection, so we kind of expected this kind of result, but it's interesting and to be able to demonstrate that and might give us further ideas for possible analysis in the future. So some possible limitations of the results that I presented here. First, this is a convenient sample. The data that I'm working with is a convenient sample. The reasons for why a specific sample has been collected from specific farms are from a specific farm is not always clear. Different veterinarians could have different procedures regarding why they are sampling specific farms, and this obviously might bias the results that I'm finding here. I do not have control over that, so I'm trying to work with the data that I have, but I'm trying to make clear that there are limitations to it. The sample composition is not always clear. Are all sequences that we are working with from a single animal within those farms or from a pool of animals within those farms? We don't have a denominator, so we only have information for farms that are positive for the sequencing. Data on animal movement is not available for our systems. And the analysis here was restricted to only a fraction of the MSHMP data. So the MSHMP data has a plethora of data that is much wider than what I used to do this analysis. And we are planning on looking into those sometime in the future. But again, there are other limitations regarding movement data, availability, specific times in which sequences were sent from different systems. So it's a choice to be made. So the take-home message that I would like to point here is that the prevalence of PRRS genetic groups is not equal in different years, in different systems, and in different production types. The occurrence of PRRS genetic groups is associated with movement of animals, or seems to be associated with movement of animals, at least for some genetic groups. Again, recording this data is important, especially if we are trying to move towards forecasting the occurrence of those diseases. And obviously, forecasting is only part of the story, because in the end we're trying to prevent those events from happening. And the PRRS vaccines are not from the same genetic group as the most prevalent sequences that we found in this data set. So I'd like to make acknowledgements, especially Kimberly van der Waal, which is my supervisor here, but also Cesar Montse and Andres, other collaborators that helped in this analysis, and especially the MSHMP team, not only from the university, but also all systems that agreed and contribute toward creating and maintaining this data set of data that we are using here. Also, an acknowledgement to the SHIC and to USDA who are funding these activities that we are holding here. Thank you so much.